Hi, and welcome to another video from OmnisRes. This video accompanies the post Half-Baked Analysis using MAF files to find neoantigens. So if you haven't already, go to our website, omnisres.com, go to our work, scroll down, and look at the post Half-Baked Analysis using MAF files to find neoantigens. So what's a neoantigen? So there's been some recent publications on this topic. For example, this one from the New England Journal of Medicine, and this one from Genome Research. I don't really know if I have permission to go over figures or data in these publications, so I won't be discussing them any further, but if you're interested, you can go ahead and look these up on PubMed and look through them. I will give a brief overview of what neoantigens are. So normally you have normal cells, and they chop up their own proteins and present these proteins as small peptides on their surface. And then you have T cells, which patrol your body, and if they see a normal cell, they just ignore it, unless you have some sort of autoimmune disease. So why does a normal cell chop up its own proteins? So when you get infected with a virus, it'll chop up the viral proteins as well, present that on the surface, and now the T cell will recognize that as a foreign peptide and kill the cell. Now, this is interesting for cancer biologists because you can have a cancer cell which will present a peptide but which has a mutation in it, and then a T cell will come along and it might recognize that as a foreign viral protein and kill the cancer cell. So how do you find these neopeptides? Neoantigens. Well, first you just take your protein sequence, the protein which got mutated, you look for the mutation, and what people do is they go 8 amino acids in both directions. So why do you do that? You do that because you normally have a monomer, so you normally have a peptide of length 9 amino acids loaded onto the surface of your cell. And if you have a mutation, you need to go 8 in both directions to get all possible monomers, because this one mutation is present in all these possible monomers. And then what some people do is they go one step further and they take that monomer, which is on the surface of the cell, and they find these neoepitopes. So it's possible that the T cell only recognizes a sub-portion of the monomer. So you might have 2 monomers, which maybe vary at this amino acid, but they both share this neoepitope, which has never been seen before. So both of those peptides would be basically equivalent in the eyes of a T cell. So what some people do is they focus on these neoepitopes instead of the monomer itself. Okay, so how do we go about finding these neoepitopes? You can download these things called MAF files. So I went to Fire Browser and I downloaded the GBM dataset. So Fire Browser is a place where they host TCGA data, and TCGA stands for the Cancer Genome Atlas. It's basically a project where they sequence thousands of patient tumors and they make the data publicly available. So you can go to Fire Browser, download different data sets, in this case GBM, and it'll give you a MAF file for each patient. So the first portion is this patient identifier, and the 01 just means primary tumor. So in this case, I think that might be a recurrent tumor, 02. So that's what the 01 or 02 means. But then, so let's just look at a MAF file. So it's a giant block of text. It's tab delimited, I believe. So let's look in Excel and see if that helps us out. Yeah, so you have a bunch of data in these MAF files. And these files come from doing whole genome sequencing on the tumor. there's a lot of information, but really we are just going to be looking at a very small portion of the information in this file. So we're interested in if the mutation is a missense or not. So if the mutation is not a missense, then, for example, if it's a silent mutation, then that mutation doesn't really have any effect on the protein sequence, so we're going to ignore those. We're also going to ignore frameshift mutations and nonsense mutations. Those are a little bit difficult to deal with, and they're fairly rare as well. We could write code for those, but it would take a little bit more time. And then we also just need to go over here a little bit and find these columns. So we're going to be needing this refseq transcript. So we need to know which transcript is being mutated. And then we need to know the protein change. So this tells you what the normal amino acid is at that position, and the position is right here. And then it tells you the change. So I actually went through all those MAF files, and I just wanted to sort of see what the data looked like. And I made a really quick histogram here. You have patients here and number of mutations. And I just wanted to see how many mutations there were per patient, and just to see what the distribution looks like. And the median was 76. And it's been reported in the literature that the number of mutations that a patient has does not correlate with survival. So I also downloaded the survival data for these patients, and I plotted that Kaplan-Meier plot. As you can see, there's no difference. So currently, this is consistent with the published literature. It's not the number of mutations that matters, but it's the specific mutations that seem to matter. And we're going to look to see how you might find those specific neo-antigens with these MAF files. Okay, so we're going to be using Python to look through these files. We're going to need the manifest file. The manifest file basically just tells you all of the names of the MAF files. Okay, so let's get Python open and let's get started here. So we're going to need to import a couple things. I always like to import at the top. We're actually going to be using BioPython for the first time. I mean, not our first time, but the first time we've gone over it. I usually just use seq a lot. So you just do bio.seq, it's from bio.seq, import seq. And I'm also going to be using the re module a little bit. And then we're going to be using a familiar tool. We did a video on parsing NCBI data, and at omnisres.com we uploaded a file called dictionaries.py, and we'll be using that in this video. Let's just refresh on what dictionaries.py does. So it's going to take a little while to load because we're loading every single human transcript. So if you do, so there's two dictionaries, mRNAs and sequence. So if you do mRNAs TP53, the second entry in the list, you get all the possible transcripts. And then if you want to know the sequence of one of those transcripts, you just paste that accession number over here, and then you get the sequence. And we also have the stop and start coordinates for each transcript. Okay, so because the MAF file gave us the accession numbers, we're going to make an accession dictionary because I don't want to have to, I don't want to have to loop through every single transcript in this dictionary. I just want to be able to call the accession number and get the information. So I'm going to make a dictionary called accession. And so for in the mRNAs, which is over here on the right, and then for J in mRNA's I, so the key, and then 1, like we just did, we're going to, I'm going to actually put a try statement. Well, I'll show you what happens. We're going to get an error, but so J1, because we're at J, and then we want the accession, and we want that to be the key in our accession dictionary. And we're going to set that equal to the start and stop. So, and we're going to make those ints. So that's just going to be J -2 and int J -1. Yep. Okay. See what happens. Yeah, so what happened here? So let's see what J is. J is a partial mRNA. It's an experimental transcript, and it has this less than 1 as a start coordinate. So that's kind of crazy. So we're going to have this try statement. So we're going to try and do this. But if you fail, go ahead and print J. Just tell me what J is if you fail. And I think we'll see that every single failure is a partial mRNA. Yep. So we're just going to now put pass here, since we confirmed that every time we have a partial mRNA, this is going to, we're not going to want that data. And these experimental transcripts aren't going to be in the MAF files, most likely. Okay, so now that we have that accession dictionary, we are ready to make a function for getting these peptide sequences. So actually what I'm going to be doing right now is I'm going to be getting this, the 17 amino acid sequence right now. So we get that by creating a function. I'm just going to call it mute, because I'm going to be mutating a protein. And I'm going to need the position of the mutation. I'm going to need the transcript. And I'm going to need to know what the mutation is. And then what I'm going to do is I'm going to return, so I'm going to make protein equal to, I'm going to now call seq from bio.seq. Basically, that just makes a seq object, which then allows you to have some built-in methods, which we're going to be utilizing. So I'm going to use my sequence dictionary from dictionaries.py, and I'm going to get the transcript. And I'm probably going to have to make this full screen, because it's going to get pretty long. And I'm going to get the accession dictionary, which we just made, to get the start and stop code coordinates. And that's also going to take a transcript. And I'm going to want the start, and I'm going to want the stop. Oh, over here, I forgot to actually subtract 1 because of Python coordinates. Python coordinates are a little bit different. They start at 0. So my NCBI coordinates are a little bit off. So I have to subtract 1 there. And then we're going to get accession transcript and the stop. Call seq on that. And then the nice thing about sequence is it has this translate method built in. So we're going to just call that. And then I'm going to return the protein. And I only want that 17 amino acid stretch. So I'm going to do position of the mutation -9, then do position -1, because I don't want to include the normal amino acid at that position. Then I'm going to add the mutation, And then I'm going to do protein, the position of the mutation, and position +8. So there's actually one problem with this code, is what happens if your mutation position is less than 9. Like if you have a mutation position at the very first amino acid, then you're going to have a negative index here, and it's just gonna mess everything up. So we need to actually have an if statement. So if position -9 is less than 0, we're going to return something else. So I'm just going to get a little tired of typing here, so I'm just going to paste that in there. We're going to start at 0 because we know that this is some negative number which we don't want. So we're just going to start at 0. And then else do this. So actually I want to just demonstrate the ability of our accession dictionaries and dictionaries.py and the translate method. So we've been using the example of TP53. So let's just go ahead and call, I'll actually print the string of the sequence object, the sequence of a TP53 transcript from up here, which we used before. I have to make this bigger too. The accession dictionary to get the start and stop coordinates. So I want to get the start, and then I want to go to the stop. So I'm just grabbing the coding sequence of this transcript by using the accession dictionary, the start and stop coordinates of the accession dictionary, and then I'm making that into a seq object so that I can call .translate, and then I'm just making it a string because the seq object sometimes it doesn't print out well if it's not a string. So as you can see, it starts with the methionine, which you'd expect, and it ends in a star, which stands for a stop codon. So let me actually just print the sequence just to convince you that we are doing the right thing here. So we start with ATG and we end with TAA. So our accession dictionary is working perfectly. Okay, so I just wanted to demonstrate that. Now back to coding over here. So now that we have this mutation function, we are ready to get some mutations. We're going to need to use that manifest file that we saw earlier. So let's just go ahead and open that. Fire browser, that's the folder, and then the file, manifest.txt. I just showed you before. It's basically, I'll show you one more time. So it's basically just this identifier and then a space, and then the name of the file. So it's pretty easy to parse. So we're going to make 2 lists. We're going to get the patients, and we're going to get the files. And those names are almost actually the same. Actually, I mean, I could use the file name as the patient name if I wanted to. So MAF patients, I'm going to append to that i.split. So I'm just going to split on the space, and then I'm also going to split on period because there's a .maf. And I'm going to take the first of that, And then I'm also going to take away some of the information, because the -01 doesn't add any information to the patient. And then for the MAF files, I'm going to append i.split 1, and that's the file. Okay. And then now that we have that set up, now we can go through all these files. So I'm going to make a peptides dictionary. And for I, j, and zip the patients and the MAF files, open the file, I'm going to need to do a double backslash here. So in Python, if you want to get 1 backslash, you actually need to write double backslash. Then I'm going to add J, which is the file name. So I have my folder name plus a slash, and then actually I think those are forward slashes. I get them mixed up. And then plus the file name. Then I don't want the header line, so I'm going to f.readline that. And then for k in the file, I'm going to take each line, which is k, and I'm going to split it. I'm going to say data equals k.split. I'm going to split on tab. And then if data 8, so that's actually the 9th column, equals missense mutation, you are going to try to call our mutation function. And this is a little bit of a beast. So we're going to get our peptides dictionary. The key is going to be the patient. So all the keys are the patients. And then we're going to do peptides.get the key. If we haven't seen the value before, the key before, we're going to instantiate an empty list. And if we have, it's going to return the list that's there. And then we're gonna have this huge thing. I'm just gonna paste it in there. So what we did here is we used our mute function from above. We looked at, so the mute function, it needs the position. So let's see how we get the position. So we have to go to that column, and we need to get that number. So what I did to get that number is I called the remodule, and it uses a regular expression which looks for any number between 0 and 9, and that number needs to appear at least once. And it looks in the 42nd column, and then it grabs that value, because read.findAll returns a list, so I need to just grab the value of the list. Then I need the transcript, which is just from column 44, Python coordinate 43, and we saw that over here. And then I need the actual mutation that's going to happen. So I need this last letter. So I could have just asked for the last letter, but I thought it'd be a little fancy and just call the remodule again and do read.split this time. Okay. And sometimes that will fail because you might not have the transcript in your dictionary. Sometimes I think they had the wrong transcript in there a couple times. So that can fail. But if it does, we're just gonna we're going to ignore it. Okay, so I think we'll go ahead and see if that runs correctly. We're not going to get an error here because we have the try except statement. So it's looking through all 200 or so files. So it is going to take a little bit of time. Okay. So let's just look at the peptides dictionary. So as we wanted, we have the patients as the keys. So I can look at one of these for ion peptides. Just take one of the patients. Yeah, so we have our 17 amino acid stretch, and the very middle will be the mutation. Sometimes you end in a stop codon because you were at the end of the peptide, and sometimes you don't because you were at the start of the protein. Okay, so now we've done really the hard part. We made the 17 amino acid stretches centered on the mutation. What we really want, though, are the monomers, which will be loaded onto the cell. So what we're going to do for that is we're going to do monomer peptides dictionary. And then for ion peptides, so basically the patients, for JN peptides I, so all the peptides for that patient. What we actually have to do is I showed you that sometimes you have the asterisk for the stop codon. So if that is present in the, it's supposed to be a 17 amino acid stretch, but if you have a stop codon, obviously it could be shorter. If it's there, now you need to make J to stop before that position. So all you have to do is remove the last position, basically. But you have to put J there. So from the start to the end, except the last position. And then once you have the correct J, so J will be the 17 amino acid stretch unless you had a stop codon. So for KL in zip, range 0, length J, -8. So I'm going to zip two different ranges together, which allows me to take slices of the 17 amino acid stretch. And I want slices of length 1. And I only want, I didn't finish my zip. I only want monomers. So it's possible that the original peptide itself could have been less than 17. It could have been really short if the mutation started early or late in the protein sequence. So I just have to make sure that I have monomers. So only if I have a monomer am I going to add to this dictionary. And the keys, again, are going to be the patients. And we're going to be doing the same strategy as before. We're going to use the get. And we're going to make an empty list, and then we're going to be adding to that list. Okay. So that was monomers, and then if you want the tetrapeptides, you just do a very, very similar thing here. You just have to go over the monomers instead of the 17 mers, and you just change your ranges. Okay, so that'll give us the monomers and the tetrapeptides. And just to save some time, I'm going to go ahead and throw some extra code in here, which will count the unique. It'll count how many times we see each peptide among all the patients. Okay, so let's run that. This code, I guess, isn't the most efficient code. Okay, so now let's actually see what let's just go through some of these non-monomer peptides, just make sure everything is fine. So again, we're just making sure we have the key as the patient, and then if we want to see the peptides for that patient, we have to use the patient's name. Okay, yep. So those are all the monomers which were affected by a mutation. And then the tetramers are going to be fine as well. So how many monomers do we have among all the patients? We have 118,000 monomers, and then how many tetra counts do we have? Okay, 88,000, not as bad. But so this is kind of crazy, right? We're trying to find these mutated monomers or these affected neoepitopes, which are tetramers, and we're trying to find specific ones which correlate with survival. But we have just an insane number. So what people do is, it's known that only a small fraction of peptides actually bind to the protein which presents them on the cell. It's known as MHC class 1, also known as HLA A, B, or C for humans. And what people do is they use software to find the binding affinity of each monomer, and they only keep the monomers which have a good binding affinity. And there's freely available software to do this. Now, where the problem comes in is there's a lot of different alleles of this protein. And so you have to know which allele you're dealing with when using the software. But this information is not publicly available. The TCGA has not recorded the HLA type of the patients. So to get this information, you would actually have to find it yourself. And what you can do is you can go to CG Hub, and you can go to the browser, and to get the HLA type, you're going to need RNA-seq. Let's see, library type. You're going to need RNA-seq or whole genome sequencing. Depends on the software you're using. And we're going to just go ahead and do unaligned. Okay, apply those filters. So we have 11,000 results. So there's 11,000 patients basically. Sometimes a patient might have multiple sequencing done, so it might not be exactly 11,000 patients. but around 11,000 patients. Let's add that to the cart and let's see what happens. This might actually take some time. Okay, so we have 70 terabytes of data that we would need to download if we wanted to HLA type all of these patients. And this is compressed data, so when you uncompress it, it could easily be over 200 terabytes of data. And, you know, at OmnisRes, we could theoretically, if we wanted to, we could download all the data, but I'm not sure how much that would cost, the server space and such would cost. It's just not quite, I'm not sure it's worth it. And plus, there's been groups who have already downloaded this data. There have been several recent papers on people who have looked through all the TCGA RNA-seq datasets for new non-coding RNAs. So this data has already been downloaded and stored. So it would be nice if these groups would go ahead and haplotype all the patients and make that data publicly available. But I'm not sure if they saved the data. They may have just downloaded it, analyzed it, and then deleted it, because storing this much data is quite a burden. What really should happen is TCGA themselves should be HLA typing the patients, and if they are too busy to do that, they should maybe, I don't know, provide read-only access to their data or something. It's just a little too difficult to work with this much data. Okay, so if you guys have access to some MAF files and the patients are HLA typed, or maybe you want to work with a subset of these patients, for example, only one cancer, then it would be doable to download the data and HLA type the patients. And if that's what you're interested in, this code should be a great start for you to start finding neo-antigens. This code will be available at omnisres.com. If you have any questions, you can leave them in the comments below or email us at omnisresnetwork@gmail.com. Okay, thank you.